Hello everyone and welcome back to AP Bio with AP Playground. Today we're covering topic 2.8, Mechanisms of Transport, and this will also serve as a bit of a review of all of the cell transport subunits that we've been covering. And the objectives here are very simple. Describe the processes that allow ions and other molecules to move across membranes. And we've been looking at this for the past 4 or so subunits, talking about the different types of transport and diffusion that can happen across the cell. But here there's a focus on metabolic energy is required for active transport. So we'll be reviewing that as well. And the suggested skill is Concept Explanation 1B, explain biological concepts and or processes. Let's start by reviewing passive transport. So the cell membrane has selective permeability. Some things are going to be allowed to go in and some things are going to be allowed to go out. And this is due to the hydrophilic and hydrophobic sections of the phospholipid bilayer which make up the membrane. And passive transport is movement of a solute across the membrane with the concentration gradient from a high concentration to a low concentration. And this is not going to require any additional energy. Small hydrophobic molecules and small uncharged polar molecules can do this themselves. They're able to slip in the cracks through between the phospholipids within the membrane, and because of their size and because they are hydrophobic or uncharged, they're able to pass through the polarity of the hydrophilic phospholipid heads. Large polar molecules are going to require membrane proteins, and this is a process known as facilitated diffusion. Because first of all, they're large, and second of all, they're charged. To be able to pass through the membrane, they're going to need to go through a membrane protein. And this is either going to be through a channel protein or a carrier protein. Channel protein lets the compound pass through. It's just like a channel. It's like a nice tunnel, and it's usually for ions. Whereas a carrier protein grabs specific molecules in specific concentrations and allows them to pass through the membrane at a specific time. And lastly, aquaporins are a membrane protein that help with the transport of water, and we're going to see this when we talk about osmosis. And this transport is going to happen until dynamic equilibrium is reached, meaning there's going to be the same concentration on either side of the membrane. Things are still going to be moving in and out. It's dynamic. However, there's not going to be a net difference. One side of the membrane is not going to have overall more concentration of a solute than the other side. So if we look in the top right at a picture, over here it's just simple diffusion passing straight through the membrane from a higher concentration of 7 cubes to a lower concentration of 4 cubes. Over here we've got facilitated diffusion with a carrier protein. It's going to use this special protein that's going to take a specific number and a specific molecule and allow it to go through the protein. And lastly, here's osmosis with an aquaporin, which is a channel protein, allowing the water to pass through the membrane even though it is a polar molecule. Next up, let's review active transport. This is the movement of solids across the membrane against the concentration gradient, from a low concentration to a high concentration, and this is going to require additional energy in the form of ATP. Membrane proteins can also be used for active transport, but this is for small amounts of molecules. For taking in large amounts of solid or molecules, we use endocytosis, which uses membrane folds. So first of all, pinocytosis is the ability to take in water or other liquids by creating an inward fold in the membrane. It's like a valley that the liquid settles into, and that closes off and traps the resource within the membrane, and that takes it into the cell. Phagocytosis is the ability to take in large molecules by the membrane reaching out and grabbing those molecules and again pinching off, forming a vesicle, and all of a sudden you've got those resources in the cell. And lastly, receptor-mediated endocytosis is endocytosis when the receptor is activated by the right molecule. So when the right ligand or signaling molecule is within the receptor, all of a sudden we have endocytosis happening. And for expelling large amounts of solid, the reverse of this, we use exocytosis, which is basically the same thing. Vesicle with the resource is going to bind to the cell membrane, and that's just going to open up and let the resources out. And again, this is all going to use energy because it is active transport. So in the top right here, we can see the use of a membrane protein in assisting moving molecules against the concentration gradient. There's a low concentration here and a high concentration there, but we're still moving in this direction. And of course, it's going to require energy in the form of ATP. Lastly, let's review osmosis. And this is the diffusion of water through aquaporins, which again are channel membrane proteins. And the amount of water is always going to try to be proportionate to the amount of solute on either side of the membrane. So water is always going to follow where the solute is. If we look on the bottom picture here, we've got an unequal amount of solute, an unequal amount of molecules in each side of this beaker. It's got a semi-permeable membrane, meaning that water can pass through, but the solute can't. So even if the solute wanted to diffuse across this membrane, it wouldn't be able to because of this semi-permeable membrane. So over here we've got a lot of solute, high concentration, over here low concentration. And because water is able to pass through this membrane, it's going to form a proportional relationship. It's going to follow the solute, and we're going to have a high amount of water on the side of the beaker with a high solute concentration, and a low amount of water, less amount of water on the side with a lower solute concentration. And this is the process of osmosis. So there are three kinds of solutions. Isotonic solution, which is the same amount of solute on either side, so there's no net water movement, meaning water can still move across the membrane, but there's not going to be a noticeable change in the amount of water on either side. Next, a hypertonic solution, which means more solute outside of the cell, which means water is going to move outside of the cell because water is always going to follow the solute. And this could cause the cell to shrink if it's an animal cell, and if it's a plant cell, the cell wall won't shrink, but the membrane inside of it will. Lastly, in a hypotonic solution, that means there's more solute inside the cell, and this especially happens in a freshwater environment, so water is going to move in the cell, and this could even cause a cell to burst. In the case of an animal cell, and in the case of a plant cell, plants actually like having pressure applied, so this is going to be kind of the normal state. And like I said, animal cells can burst or shrink in non-isotonic environments, and some use what's called a contractile vacuole to pump water out, which is basically going to be pumping the water out as it comes in order to keep the cell alive, and it'll do this until it's reached back to an isotonic environment and doesn't need to function any longer. Lastly, water is going to move from a high water potential to a low water potential, meaning a place where water has a lot of free movement. It can bind to molecules if it wanted to a place where it doesn't, meaning it's going to go from a place without much solute to a place with more solute. It's going to go from low osmolarity to high osmolarity, less solute to more solute. Lastly, lower solute concentration to higher solute concentration, less solute to more solute. So these are just some terms you may see on the exam. Remember the difference between hypertonic and hypotonic solutions, and don't get confused. And just remember, water is always going to follow the solid. So here's the mechanisms of transport review. Just know your passive and active transport, know your osmosis, and these topics weren't covered in full depth in this video, but basically all of 2.5 through 2.8 cover these topics more in depth if you'd like to take a look at those. We'll see you in the next one.